Thank you, Kaleida. I would welcome everyone. I'm very pleased to be able to present in this wonderful seminar series. Thank you so much for inviting me. It's a great honor and pleasure. And my goal is to spend the next about 45 minutes to tell you a little bit about the thermodynamics of ternary asymmetric lipid membranes, a topic that I've been very interested in over the past couple of years. Now, there's lots of weird words in there, mixed and thermodynamic and asymmetric. So I spent the first part of my talk to introduce the two subtopics before I dive right into combining all of these things. So, okay, so let's see, where do I start? Turns out, over the last couple of years, I've been working on a very famous flag. That flag being this one. It's a flag for asymmetric membranes. It turns out that you cannot really go to any talk on asymmetric lipid membranes without somebody showing this picture from the wonderful paper by Joseph Laurent, spearheaded by the Leventhal group. And so what is this about? It's about asymmetric cell membranes. So let me give you a brief introduction to cell membranes and then go right into asymmetry. So if you, let's say, open a book on molecular cell biology, very early on, you're going to see a picture, something like this, a cell cut open, and most anything you see are membranes, the outer envelope, the ER, the Golgi, all of these things. So if you look, for instance, at the outer membrane a little bit more closely and say, oh, let's zoom in there, you say, oh, yeah, okay, so actually you can't see anything because the JPEG isn't very good enough. But if you were to be able to see a little bit more, you say, ooh, a membrane, very nice. And if you zoom in even more, you then realize that what we're talking about are these funny things, bilayer of lipid molecules with proteins chucked in there and like sugar molecules and cytoskeleton elements on one side and extracellular matrix on the other side, something really very complicated. And rest assured, I'm not going to even come close to that level of complication in today's talk. What I will mostly focus on in today's talk is these guys, which are the lipid molecules that make up these membranes. And then in a good physicist fashion, we would then illustrate this as like something like this, lipid molecules forming a bilayer leaflet, and they're all standing next to each other, they're all self-assembled. This, of course, is a little bit too simple because this looks very static and very crystalline. At the very minimum, this is actually a disordered fluctuating system. This is a 2D fluid. That's still a little bit too simple because this is just one lipid species. This is what we've investigated in the lab for like 50 years, but nature is more complicated than that. For instance, nature have lots of different types of lipids, as illustrated by the different colors. And it's even more complicated than that because the lipids are not just randomly here and there. There seems to be order in the sense that one of the two leaflets compared to the other leaflet has different types of lipids in it. And so here, for instance, also illustrated there's cholesterol molecules that are preferentially on one side and not the other. And so at this point, we are coming close to what actually is a biomembrane, except minus the proteins and all the other complications. But what we have here is a two-dimensional self-assembled system of lipids that then would host the proteins as it was originally proposed more than 50 years ago in the famous fluid mosaic model, which says, okay, it's the fluid bilayer in which the proteins are embedded like in a mosaic. Now, interestingly enough, it turns out that that there's another paper from basically the same time which said, and these bilayers are asymmetric in the sense that the lipid composition on both sides is not the same. And this paper is basically just 12 days later. So basically from the time on that we know what the structure is of a lipid membrane, we also know it is probably asymmetric. Turns out, just a year afterwards, a first paper got published that tried to quantify that asymmetry. And this paper came from the Netherlands, from the group of Lawrence van Denen, who showed, looking at the human red blood cell, what types of lipids, SVP, C and P, these are sphingolipids, phosphatidylcholine, phosphatidylethanolamine, various types of lipids, how many of them are on the outside of the red blood cell and how many are on the inside of the blood cell. And this is sort of what they proposed is probably going on. Now, in that famous FLAG paper from a few years ago, this was also remeasured with a lot more detail, and it's in color, so it must be new. It also turns out it looks very different. The reason why it looks very different is because it's plotted kind of differently. If I can actually make this look a bit more simple by saying, first of all, the new authors used a slightly different normalization. So let's use the normalization that Van Denen used back then. Also, the new authors could distinguish things which back then were hard to distinguish. So let's go to the situation where we combine things that back then weren't distinguishable and order it in the correct way. If we do that, then the plot starts to look extremely similar to what was measured back then, and tells you that 50 years ago, these people were really very good at measuring lipid composition on a side-by-side basis. There are differences, and these differences are, as always, subject to a lively debate, which I'm going to skip. Suffice to say, of course, that we have now an awful lot of information, for instance, from this paper about what kind of lipids are on which side, and what kind means specifically what lipid chemistry or lipid head groups, what's going on with the tails, what's about tail saturations. There's a lot of information. Okay, that's all fair and square. However, it turns out that there's more to life than composition. There's other things in a membrane that are different on the two sides. And the thing that I would like to spend a few minutes talking about is something that I call differential stress. So if we go back to this particular membrane, then it turns out we have two leaflets, and these two leaflets are subject to individual lateral tensions that I'm going to call sigma plus and sigma minus, and they don't have to be the same, it turns out. The moment you break the symmetry of one particular observable, as a physicist, you know you're probably going to break a whole bunch of other symmetries in the process. So if that's the case, and if we have two different surface tensions, sigma plus and sigma minus, it turns out for logistic reasons or for like reasons of ease of dealing with the system, it is useful to, rather than dealing with the individual surface tensions, to instead take a linear combination of those two, which is the sum and the difference of these two surface tension. I'm going to call the sum the total tension and the difference the differential stress. This is nothing particularly mysterious. All I'm going to do is I'm going to take this two-dimensional space of possibilities of tension and pick a different axis set in which I'm describing this, which is the two diagonals, the sum and the difference. Why do I do that? Because it turns out that in a sense that I will define a little bit more carefully later, the overall net surface tension often turns out to be very close to zero, while the differential stress is the variable that is actually large and dominant. And I'll see in a second what that means. So basically we have now one particular variable, delta sigma, that explains the axis along which a lot of the relevant physics is happening. So given that, what do we know about this differential stress? Unfortunately, turns out very little. The reason why we know very little is because it is really very difficult to measure leaflet tension on membrane tension on a leaflet by leaflet basis. So people are working on this, and there's good ideas that they have about how to do it. But at the moment, this is just a puzzle. Given that, why am I talking about things that haven't been properly measured yet? The reason is that there are indirect reasons that supports its existence, in the sense that basic physical consideration would set a natural scale for the value that this variable has. And I'm going to give you two examples for a physical reason that points towards the existence of such a differential stress. The first reason comes about when we start to think about membrane curvature, membrane bending, in particular membrane torque balance. What I mean by that is, let's take a piece of an asymmetric membrane which has some lipid species on one side and another lipid species on the other side. Because it's different lipids that have different properties, maybe different intrinsic curvatures, there is a natural bilayer spontaneous curvature that such a piece of bilayer would have, in the sense that its curvature energy isn't zero when it's flat, it's zero when it takes this value. And so if we write the bending energy as 1/2 times the modulus times the difference between the curvature and the spontaneous curvature squared, then the derivative of this bending energy with respect to the curvature is what's called a torque. That's what's bending the membrane. And if I, for instance, evaluate this torque for the flat state, k equal to 0, then I'm going to get basically the product of the bending rigidity and that spontaneous bilayer curvature. So that's what wants to make the membrane bend based on lipid individuality, lipid shape, if you like. But this is not the only reason why a membrane would want to bend. You could alternatively look at a membrane that has the same lipids on both sides, except they're differently densely packed. So on the upper side, for instance, they're more closely packed, and on the lower side, they're less closely packed. So there is a force on the upper side that would like to push the lipids apart, and there's a force on the inner side, presumably it wants to pull them together. And since this force you could think of as acting at some suitably chosen reference position within the bilayer, I mean, just say the middle or something like this, that we call that Z0, then the product of the individual tension multiplied by basically a lever arm, Z0, creates a couple, and that is essentially a torque, which derives from the differences in these tensions. And it then turns out that if you take the sigma plus times Z0 minus the sigma minus times Z0, you get the differential stress times that reference surface, like neutral plane or something like this, which also creates a torque in the system. So we have two independent reasons why there is a torque in the system. Okay, so how can we make this make use for us? So here's an example. So what I'm showing you here is a picture that was one in the early papers where people managed to create artificially asymmetric lipid bilayer vesicles. This is a vesicle, for instance, where the authors tell us the outside consists of a lipid that is called POPC, and the inside consists of a lipid that's called DOPC. Now, we know enough about these particular kind of lipids to look up in various tables that a system compositionally asymmetric like that should have a spontaneous bilayer curvature of about 0.017 whatever inverse nanometers. And so 2 divided by that curvature would be the radius of a vesicle, depending on the energy of which would vanish. Okay, but that's just a few tens of nanometers as opposed to, this vesicle has a size of 20 microns, so which is 2 orders of magnitude bigger. Why does this vesicle not immediately spill out little tubules that have the radius that the membrane prefers? How can we make membranes that are the curvature of which is 2 orders of magnitude smaller than what the actual material prefers? So the reason, I think, is that we don't want to set the torque based on lipid curvature to 0. We want to send the entire torque to 0. And the entire torque consists of the differential stress part and the bending part. So if I set this piece to 0, then I can solve this actually for delta sigma. And it turns out that all the other variables in there, the bending rigidity, the spontaneous curvature, and the reference surface have values that I roughly know what they are. The bending rigidity is about 30 kT, K0b, I just told you what it is, and Z0 among friends is a nanometer roughly, right? Plus minus a little bit. And if you plug in the numbers, you get something like 2 millinewtons per meter. Now, if you're not really deeply into the membrane field, 2 millinewtons per meter doesn't mean a lot to you. So I'm telling you, this is between 1 and 2 orders of magnitude bigger than any typical membrane bilayer tension that you will typically talk about. It's also very uncomfortably close to the bilayer rupture tension. So if you maybe double that and apply it to a bilayer, that bilayer might start to fall apart. That's the sense in which I meant that the differential stress is very much bigger than the individual, the total tension in the membrane. Let me give you a second example for why we might expect a differential stress. And so let's start with a membrane that consists of 2 types of lipids, so it's really compositionally asymmetric. And they're done in such a way that, let's say, the upper leaflet is polyunsaturated lipids, lots of double bonds, lots of disorder. The lower leaflet is saturated lipid. And let's also say we kind of arrange the situation cleverly in such a way that this particular membrane actually doesn't have any differential stress. And let's now add cholesterol. This is interesting because cholesterol is very well known to prefer saturated lipids when it has to decide where to go. It very rapidly flips between the 2 sides and it picks the side that it wants to go to. Now, if cholesterol prefers to go on one side and not the other, it will squeeze the lipids on one side more than on the other side. And so you would imagine because of that, because of this asymmetric distribution of cholesterol, you now create a stress difference between the 2 sides. And you should, okay, so I mean, how big is that going to be? That turns out not to be quite as easily calculable than the example I gave you before, but it's in principle at least clear what kind of theory you would write down. You would write a theory that contains things such as elasticity, the stretching of the membrane, the entropy of cholesterol, and some preferential partitioning. And a couple of years ago, I had done this with a student of mine, Malavika Varma, and we've come up with a prediction for what this should look like. And then putting in reasonable parameters, you get a curve such as that, the differential stress, as a function of the mole fraction of cholesterol in the system. For 0 cholesterol, the differential stress is 0, mostly because that's just how we set up the system. And the moment you add it, the differential stress very rapidly shoots up because there is a preferential distribution of cholesterol going into the saturated leaflet. Then if you keep adding cholesterol, at some point there's so much cholesterol in there that the membrane again becomes essentially symmetric because most of it is cholesterol, so it goes down again. But for real membranes, the ones in cells that we care about, the mole percent of cholesterol is somewhere between 20 and 40. And if I just plug in this number, then I'll end up getting again at that magic number, 2 millinewtons per meter. The fact that it's exactly the same is a total coincidence, but a few millinewtons per meter is a number we should bear in mind as a reasonable expectation for what this kind of differential stress should probably look like. Okay, and so that's, I guess, what I wanted to say about differential stress. Now, it turns out there's another flag that many people over the past more than 20 years have been working under. Me, not as much as others, but here's another flag that gets everyone excited. I picked this particular picture for that flag. And it's, of course, the ternary mixture of membranes. So this picture was taken by a very famous paper by Sarah Veach and Sarah Keller, but there's other groups that worked on it, like Jerry Feigenson or Evan London. There's a large amount of research about the phase behavior of ternary lipid membranes. And we're going to do this a little bit of injustice by summarizing all of this in one slide, why we care about this. The reason why we care about this is that, well, ternary mixtures that consists of saturated lipids, like no double bonds, unsaturated lipids with a couple of double bonds, and cholesterol, show a number of different phases, including two fluid phases of different order and the coexistence regions of those, which terminates in a critical point, suitably chosen temperatures, right? And this so this simplified model now turns out to be a remarkably good proxy, we believe, for much more complicated biological membranes. Not, of course, in the sense that biological membranes would be ternary mixtures. They're much more complicated. There's like hundreds of lipids in there, but in the sense that biological membranes also seem to show, under suitable conditions, the coexistence of two fluid phases, which terminate in a critical point. And so not lipid by lipid, but phase by phase, there is an analogy between this system and the real biological membrane. So this is why this has been studied a lot. And so since I would like to show what these phase diagrams look like, I will now have to give you a quick 2-minute rundown for how to actually plot phase diagrams of ternary mixtures. So here is like the real estate for that phase diagram. It's a system, it's a triangle, it's called the Gibbs phase triangle, which has 3 corners, which are labeled C, U, and S for cholesterol, unsaturated, and saturated lipids. These 3 components, since they make up the whole membrane, their percentages have to add up to 100%. And that constraint means that we don't actually have 3 degrees of freedom. We only have 2 degrees of freedom, and which is why we can plot it in a 2-dimensional phase space. The way this is plotted is you just pick, let's say, a point in here, then you draw these 3 lines, and you say, all right, where these lines hit the axis, you can say, all right, we have about 50% along the unsaturated axis, 20% along the saturated axis, and 30% along cholesterol. So these are the fractions that I'm adding. And they do, in fact, add up to 1.5 plus 0.2 plus 0.3 is 1. And you can see this because the length of these arrows, in fact, does add up to the whole length of the triangle. And this works for whatever point in the triangle you pick. And so this is a way of illustrating what the compositions are for the system. It's also useful to know that if you go closer to a corner, the composition gets more and more dominated, but whatever, is at that corner. And if you go onto one of the sides of the triangle, then what you're looking at is a binary mixture, in this case, for instance, between cholesterol and saturated lipids. Okay, so this is how we plot it. Now, what does commonly a phase diagram of such ternary systems look like? So the stereotypical phase diagram here looks a little bit like this. So there's a lot going on here. First of all, what's going on here is that there's this corner up here where if you add more than 66% cholesterol, you can't chuck it in the bilayer. It crashes out of the bilayer. So points up here do not just correspond to a bilayer, they correspond to bilayers in coexistence with cholesterol monohydrate hanging out in solutions. So we have to stay below that if everything is supposed to be in the bilayer. We then realize if we go to a side, let's say the lower side between unsaturated and saturated lipids, there's a large miscibility gap because these don't happily mix. There's a coexistence between a fluid membrane phase and, in this case, for instance, a gel phase of saturated lipids. A similar miscibility gap also coexists on this side between saturated lipids and cholesterol. Curiously, it doesn't exist on the other side between cholesterol and unsaturated lipids because of the temperature that I chose, they mix at any proportion. In the middle of this phase diagram, there is a 3-phase coexistence where all 3 phases, three phases coexist at the compositions of the corners of this triangle. And the sides of this triangle touch the 2 coexistence regions that we've already seen. They also touch a 3rd coexistence region, this one, which doesn't touch the 3rd side of the triangle because everything mixes on the cholesterol unsaturated side, and which means this 2-phase coexistence region has to terminate on its own. And it does so by terminating in a critical point where a liquid disordered and a liquid ordered phase become identical. So this is a really fantastically curious phase diagram where there's a lot of fun physics going on that's been studied for a long time. It will turn out for the future, I will look at phase diagrams like this also computationally, but this is too complicated. We've developed a model with a very highly coarse-grained with our very highly conjugated lipid model, where we qualitatively reproduce this phase diagram, but not in its entirety. We don't get the gel phases really. We don't get the three-phase coexistence. We do get the thing that we care about, which is the fluid-fluid coexistence of an ordered and a disordered phase that terminates in a critical point. And we're going to use this particular model that we recently published in order to illustrate a few examples of what I'm going to say in the future. So, and with that, I've given you enough background to understand what the title of the talk is going to be about. So what do I want to do? The goal for today, and actually for my group, probably for years to come, is to figure out how these two pieces of physics play together. What if I have ternary mixtures, but in membranes that are asymmetric? So what can we say about this? So spoiler alert, at the moment, I don't have an awful lot of results yet. This is a complicated thing and will probably keep me busy for a while. I'm going to show you some small things, but not yet big results. However, I think what I can do, because we thought about it long enough, is to give you some hopefully useful ideas of how to even think about these systems. What are even the proper axes that we're going to plot in phase diagrams when we study these systems? Okay, so let's go into this. Superficially, this doesn't actually look like such a complicated scenario. We have two sides, both sides are ternary, so I guess we get two Gibbs triangles. The one triangle with this blue dot for the upper leaflet and another triangle with this magenta dot for the lower leaflet. And you have these two compositions. And since I don't want to draw two triangles, maybe we can just put them together. We have one Gibbs triangle and 2 points in there which tell us what these compositions are. Okay, fine. So why is this difficult? Then you also say that any of these points can just move around in this phase diagram. So the first has like 2 degrees of freedom moving up and down and left and right. The second one, so overall 2 plus 2 is 4. We got 4 degrees of freedom in the system. Or do we? Is 2 plus 2 really 4? Is this the right way to think about this? Turns out it's actually a little bit more subtle than that. And the reason why it's a little bit more subtle than that is because we have complications arising now. The moment we put these two leaflets, these two ternary leaflets together, one of the components is cholesterol. And cholesterol very rapidly flips between the leaflets, and we can't just put it in one and then just hope it stays there. What instead will happen is that the moment the cholesterol fraction in one leaflet touches the cholesterol fraction in the other, they start to rapidly exchange back and forth until the chemical potential of cholesterol in the two leaflets is equilibrated. And that condition, that the chemical potential in the plus leaflet is equal to the chemical potential in the minus leaflet, is a constraint that removes a degree of freedom. So we actually only have 3 degrees of freedom. However, 2 plus 2 is 4 because there is another degree of freedom which we haven't seen yet and which actually is invisible in this diagram because the two components, fraction of C, U, and S, are only relative fractions. None of the corners tells us about the total number of lipids. So what we're missing is information about the abundance of lipids in either one of the leaflets. And that lipid abundance, which I'm going to measure by the difference of the phospholipids in the upper versus lower leaflet divided by the sum, this abundance asymmetry, so if this is not 0, then it's abundance asymmetric, is another degree of freedom, which is the non-compositional aspect of asymmetry and the one that this phase diagram doesn't show us. So we have to reason about it a little bit independently. So how does this work? First of all, what are good choices for these degrees of freedom? One choice is relatively obvious. I will probably want to tell for both leaflets what fraction of lipids is saturated. I'm going to call this saturation ratios. In the plus leaflet, how many saturated lipids compared to all the phospholipids do I have? And the same in the lower leaflet. And compositions with the same saturation ratio end up being on these lines that go through the cholesterol corner and whatever down there on the US axis you're going to hit. So that's fairly easy. So we had these lines. And now we need to know where on these lines are we. And so you could say, well, we just have to tell how far up and down you are. Maybe you could just give the cholesterol fractions. And that's, of course, entirely okay, except the cholesterol content is nothing that you can easily experimentally tune because it determines itself on its own. So it's probably nicer to use a degree of freedom that's actually controllable. And what's controllable is the total amount of cholesterol in your system. So let's say cholesterol content between all the saturated, unsaturated, and cholesterol lipids. So this is something I can, but how much do I pour into my system? So that's another degree of freedom. And the fourth one better be the non-compositional one. It's that abundance asymmetry. So now I have 4 degrees of freedom, which can describe this system. Of course, the question still remains, now where on these lines are our 2 points going to end up? So I have to somehow figure out how does phi and alpha determine the location of these lines? Well, how does it happen? Of course, there is a cholesterol fraction which you can read off on the if I go to the right-hand side. So how is this determined? Well, think about it. What if I, for instance, add cholesterol to the system? If I do so, and since it can flip easily between the two sides, what will probably happen is that on both sides, the cholesterol fraction is going to go up, right? In the sense that the way that the compositions in both leaflets changes is in some parallel fashion. They might not go up by the same amount, but they will both go up if I add cholesterol to the system. Now, this is very different if I instead change the abundance asymmetry, because if I cram more lipids into the upper leaflet, that's going to squeeze cholesterol out of the upper leaflet. And it has to go somewhere, so it goes into the lower leaflet. So now we get some kind of anti-parallel motion. I increase the abundance symmetry, more phospholipids, relatively speaking, in the upper leaflet, and I get this particular motion. So this is interesting. I get two very different responses to alpha and phi changes. What that means, we can now tune the cholesterol concentration phi plus and phi minus independently. The way this works is, imagine you change phi and you change alpha by small amounts, delta phi and delta alpha. Then for small changes, there's probably going to be some linear response that tells you how the phi plus and the phi minus change in the individual leaflets. And so there's some matrix that describes this. And for the phi plus, it's going to be these two components that describe this. And for the delta phi minus, it's going to be these two components. But remember, there's parallel and anti-parallel motion going on here. So in this matrix, there's a very conspicuous minus sign in there. And because of that minus sign, this matrix is invertible. So if you tell me what kind of delta phi plus and delta phi minus you want, I'm going to just take the inverse of this matrix, multiply it on the left-hand side, and that will tell me how I have to change the cholesterol content and the abundance in my system to exactly get these 2 points. What that means is essentially that you can actually tell me what kind of points in this triangle you want to coexist, and there's going to be a phi and an alpha that will make that happen. So actually everything can coexist, provided you can play with cholesterol and abundance asymmetry. Okay, so let's say I have these 2 points, and the magenta point and the blue point happily coexist. Of course, I just said I can make many of these points coexist. I just have to adjust the abundance asymmetry. But all of these points, even though they chemically coexist, will have different mechanical properties. They might differ in the amount of differential stress, or more importantly, they might differ in the amount of torque that this membrane is under. And so among all these points, there's probably one special point where the torque happens to be 0. And if that's the case, this particular special point, I can make stable GUVs, giant unilateral vesicles, because they want to be flat. So that I have to get the torque to 0. So let's say it's that point. So these 2 points now not only chemically coexist, they also mechanically give rise to a stable bilayer that wants to be flat, and therefore I can make giant unilateral vesicles out of this. Of course, this argument works for whatever the saturation ratio is on the blue side. And so for every one of these blue lines, I can find a point that exists at 0 torque. And that tells me that there's actually a line, a 0 torque line. Every point on that line in the blue leaflet would chemically and mechanically coexist with that magenta point on the other side. But that, you see, you cut down a degree of freedom. I cannot make any point in the triangle coexist with a magenta point, only the ones on that line, if I want to make GUVs out of these systems. And of course, this argument I can make for every point on the magenta line in the other leaflet. And so I get a whole number of curves in there, which I call fancily a torque foliation, that tells me if I pick something on the one line, where am I allowed to be on the other leaflet in order to be able to make proper vesicles that just don't fall apart on me. Okay, so that's interesting. You lose a degree of freedom if you insist that the torque vanishes and you can make giant unilateral vesicles. Okay, so if you have these 2 points, you coexist, and now you do a perturbation. Let's say you pull cholesterol out of the system, then presumably both of these points move down to lower cholesterol concentration. But there's nothing that will guarantee you that when they do so, they still end up on the corresponding torque foliation curve. They might end up on different curves. And if they do so, the system after that perturbation acquired a torque. And if it acquires a torque, the GOVs might now be unstable, and they might change shape, or they might spit out tubules. This is even more obvious if I do an abundance asymmetry, because then definitely one point is going to move up, the other point is going to move down. They clearly cannot stay on the same 0 torque line. So a torque develops and the GOV might tubulate. That tells you that if you're interested now in making these vesicles and then perturbing them by adding cholesterol, adding phospholipids to one side, how does the system respond? And so as physicists, we then dutifully define response functions, such as we call them susceptibilities or something like this. Like how does the differential stress, delta sigma, or how does the torque, tau, change as I change the cholesterol fraction phi or the abundance asymmetry alpha, keeping the respective other one constant? What happens here, right? And then we want to talk about them, so we have to give them names, and that turns out to be the hardest part. So this one you could call isocompositional differential stressability, which is a mouthful, and I don't think it's a terrible name. I need help. I need names for these four friends, and I don't know how to call them and talk about them, but this is now high priority. But let's think about these things. One nice way of how to make sense of this is to just say, okay, let's focus on the torque. If I change abundance and cholesterol content in a membrane where I've picked, let's say, the saturation ratios, what torque would happen? So here's an example of something where we measured this in a simulation and then fitted a nice flat surface, smooth surface to the results, and you get the torque in the membrane as a function of, on the horizontal axis, how much cholesterol total is in it, on the vertical axis, what's the abundance asymmetry of the phospholipids. And you realize, well, sometimes it's positive, sometimes it's negative. There's this line in here where the torque is zero. This is the zero torque line. So those particular compositions, I probably will be able to turn into GOVs because this thing will stay flat. There's another weird curve up here. This turns out to be the one where the differential stress is zero, which is completely different condition. There's also this dashed line in here, which is a torque null line. This is the place where the torque is insensitive to changes in the cholesterol. So lots going on here. And that, in fact, reminds me, these susceptibilities really are just crosscuts through this surface, and we just ask what's the slope. If you imagine this is a surface over phi and alpha, I cut it at constant alpha, I'm asking what's the slope in the phi direction. That's one of these susceptibilities. I could do the exact thing the other way around. How does the torque change with alpha as I fix phi? And that's the other susceptibility. So let me, in one example, show you what they look like. So here's an example of like d torque by d phi. The horizontal axis changes phi, the vertical axis is now that slope. You realize you get, turns out to be lines in this case. Sometimes you get negative susceptibility, sometimes you get positive one. There's a particular point. These individual curves correspond to different alpha values. Here's the torque null line where the sign of susceptibility changes. This dashed line is where the torque is zero. So if you focus on only the zero torque states, then the susceptibility is always negative. Anyways, so those are these things. Another question you could ask is, well, let's say we picked fairly carefully the saturation ratios on the two sides to be not too far from what people think is going on in the human, let's say, red blood cell membrane, or what corresponds to this thing. Then I also know that in the human red blood cell membrane, the cholesterol fraction is about 40%. If I then say, okay, so maybe the torque is also zero and that's why it's stable, what's the abundance asymmetry in the human red blood cell membrane? Well, I can just go up here and go to the zero torque curve and go to the left and say, oh, it's not zero. There's an imbalance in the number of phospholipids, which in these units that I'm measuring is about 3%, and the outside leaflet is depleted of phospholipids. Turns out this is qualitatively in line with what people find, but it's quantitatively very far off. Because quantitatively, there's a recent paper where they say the difference is not 3%, the relative difference is about 20%, which is enormous. So we don't get 20%. Why don't we get 20%? Well, it could very well be that our model is just too cheap to do this, because mixing cholesterol with anything is actually a very complicated thing. Or the experiments are wrong. It's probably our model that is wrong. But if nothing else, it tells you that the 20%, which not at all voluntarily comes out of a model, is a very strong and very remarkable statement about the state of affairs of our red blood cell membrane. This is massively asymmetric and would be curious, like, why is that the case? So towards the end, let me show you two applications where we found out a few things. So let's take a system with this. Here's the phase diagram measured from our model, and let's pick 2 points and make them coexist. On the lower leaflet, the magenta point, and on the upper leaflet, the blue point. I picked the blue point very far away from coexistence, and so this is a nicely mixed system, but the magenta point is fairly close to the phase coexistence. So this is not yet quite in the coexistence region, but this is at the brink of wanting to phase separate. And now I can do things. I can now change, without adding any liposomal, I can change the abundance asymmetry in the system. Let's say we do this, right? We increase the abundance asymmetry. Then what happens is I squeeze more phospholipids into the blue leaflets. Therefore, I squeeze cholesterol out of the blue leaflet. And so it goes into the magenta leaflet and move that point further away from coexistence. But I could do the opposite, right? I could reduce the abundance asymmetry, and then the cholesterol is being sucked out of the magenta leaflet, and so the magenta point might now plunge into the coexistence region. Could I drive phase separation purely by abundance? And the answer is we can. So here's an example exactly of these three states. The middle is the one that we started off. These are just schematic pictures of the phases that I see in the lower leaflet. Red and blue corresponds to sort of the unsaturated and the saturated lipids, and open circles versus solid circles correspond to what a hidden Markov model would identify as a liquid disordered or liquid ordered phase. So we see in the minus 10% abundance asymmetry, we start to see a liquid ordered domain that just hangs out there and stably exists, which becomes a lot more flaky, and for alpha equal to 0 and for alpha equal to plus 10, it's basically melted away, and there's no domain there. So we can drive the creation or the melting of domains purely by abundance and by differential stress. And then we said, oh, this is really cool. Maybe we can do this in curved systems. So let's try to do this in curved system. We take our system, we again pick a point somewhere here, which is like not in the coexistence region. So these should be mixed systems, but let's not do this for flat membrane. Let's do this with these kind of lipids for a curved vesicle. And actually, let's permit not just the cholesterol, but all the lipids to change. So to flip between sides, so that we say, if we wait very, very long and every lipid decides where it wants to go, maybe because these lipids might have different spontaneous curvatures and the curved vesicle provides real estate with different spontaneous curvature, maybe the system will now order the lipids across the two different sides. So we ran simulations doing this, and we found, oh yeah, there is some interesting phase separation going on. So we started at that cross where both leaflets have exactly this position, this composition, and then it turns out, all right, the inside depleted of cholesterol, the outside, the inside increased in cholesterol, the outside depleted of cholesterol, the inside sucked up more saturated lipids, the outside depleted saturated lipids, the inside ran into the coexistence region and phase separated there, and the outside didn't. So that's interesting. So you have a system which, without you doing anything, spontaneously became asymmetric because there was a drive for phase separation. You were close to a phase separation region, but the curvature helped you to sort it until something happened. So we said, that's great. Let's get statistics over this. Run a second simulation. You run a second simulation, and then this happened. So again, they ran apart. The inside increased in cholesterol, the outside decreased in cholesterol, but now they ran exactly the opposite way. The inside now depleted of saturated lipids and the outside increased in saturated lipids. And since the outside didn't quite make it all the way to the coexistence region, the outside also didn't phase separate. So both of these systems are still purely mixed and exactly opposite in their asymmetry that they achieved, which is what? So we were very confused about this. Clearly, there's something to be explained here, and we were puzzled. We just spent a lot of time figuring out whether our code was wrong. So where does one go from there? Well, the future is we really have to do some theory about this. We have to write some coupled free energies of two ternary systems. We couple them elastically, thermodynamically, figure out how to throw in the curvature so that you feel if there's a biasing force. That needs to happen. Once local compositional couples across the leaflet, this becomes very important because you might have domains. Do they register? Do they enter-register? That needs to be in there. And of course, phase coexistence becomes very tricky then. So we haven't done that yet, but my students are starting to work on this. Good luck to them. So stay tuned. At some point, I will be able to tell you what comes out of this. And if there's anyone out there who likes to do experiments on these asymmetric systems or simulations or whatever, help me work on this and talk to me. I think this is super exciting, and I hope to make some progress on this. And that's all I had for today. Thank you very much for listening to my ramblings here. And again, many, many thanks for the BPPB organizers for organizing this. And if there's time left for questions, I'd be very happy to answer them. Thank you.